Hello again, students. Can you hear me? Yes. Okay. Thank you. Today, I'm going to teach you some lessons about DNA replication in prokaryotes. Okay. We want to talk about DNA replication. Some of the lessons you studied in your bachelor degree. So if you think I repeat it and you know about it, please tell me to go further. Can you repeat polymerase? Okay, I will talk you today. Okay, Fatima. I will talk about it today. Another questions. Is it better for you? Is it better for you to have makeup on Fridays or Sundays? Friday or Sunday morning? Okay, no problem. I told you I will repeat it. Okay, don't worry. You want to have makeup Friday? Okay. So, we have one of you say Friday, one Sunday. No Sunday have 4 lecture. Okay. Friday morning, 8 in the morning, we will have makeup. Okay, 8 in the morning. Friday. Okay. Thank you. Thank you so much. So, lecture. Okay, just listen to me. Just like one lesson, one lesson. I think about 1.5 hour. 1.5 hour. 1 hour and a half. Okay. Listen to me and please record my voice. So if you don't understand, you can return to my voice, okay? And listen to me. In DNA replication, the direction, can you tell me please the direction of replication? Can anybody tell me the direction? What is the direction of replication? Is from 5' to 3'. Okay? Every time. Listen to me. Remember, every time the replication direction is from 5' to 3'. In prokaryote, in eukaryote, in leading strand, in lagging strand, everywhere, the direction of replication is from 5' to 3'. Okay? Don't make mistake. Anytime the direction of replication is from 5' to 3'. All Yes. Okay. I think we have problems because the dawn was disconnected for me. Okay. So my voice is clear. Okay, thank you. No, this is not in that book. I will give you my slides, okay? Next session, I will give you my slide too. So I was talking about the direction of replication is from 5' to 3'. When the two strands of DNA separate, we have the part, we call it replication fork. We call it replication fork. And two mother strands separate from each other, and the daughter strand, look at here, look at my pointer, from 5' to 3', from 5 prime to 3 prime synthesize. Okay? Look at this. At first, we had 3 theory for replication. One said the replication is dispersive. What does it mean, dispersive? Dispersive theory said that we have just one maternal, one partial maternal DNA and one daughter. No, one part maternal DNA, one daughter DNA, one mother DNA, one new DNA. You can see here, one part is new DNA, the red one is new DNA, and the gray one is older DNA. So the dispersive theory said that in DNA replication, we have one part new and one part old DNA. We had another theory, it was semi-conservative. What does semi-conservative mean? Semi-conservative means that we have one older DNA and one new DNA. It means we have one strand of mother DNA and one strand of daughter DNA. We have another theory. It was conservative theory. What does conservative theory mean? Conservative theory means that from one double strand older DNA, we have double strand new DNA. So we have double strand new DNA from double strand mother DNA. So you all know which theory is correct? Which theory is correct? What is the replication theory? How does our DNA replicate? B. Yes, of course. The semi-conservative is correct, so the dispersive and conservative theory are incorrect. Yes. Of course. There was an experiment that showed the B is correct. What does that experiment? At first, they put the E. coli, in the culture, in the medium, which has nitrogen 15, isotype 15 of nitrogen, and then they transfer the bacteria to the medium which had nitrogen 14. Okay, so at first they culture the E. coli in nitrogen 15 medium, then they transfer it to nitrogen 14. After 20 minutes, what does it mean 20 minutes? It means after 20 minutes we have one cell division. Yes, after 20 minutes they extract the DNA and they put it to the density gradient centrifuge, and they showed we had only one bond, which is combination of 14 and 15 DNA. Okay, and after 40 minutes, which means we have two cell division, they extract the DNA and transfer it to density gradient centrifuge, and they show two bonds. One bond is combination of 14-15 and one bond is combination of 14-14, means new DNA and one combination of new and old DNA. What do you understand from this result? If anyone can tell me, I will give her or him a positive point. How can you or what you understand from this result? I will give you positive points. No, Mr. Haydar. I told you this is the experiment that we want to show which type of replication is true. We didn't know before. We didn't know our replication is conservative, semi-conservative, or dispersive. This is the experiment which shows us which type is correct. You all know the B is correct, yes? The semi-conservative is correct. We all know the semi-conservative is correct. But how does this experiment show us the semi-conservative is correct? Who can tell me? I will repeat. Another more. Listen to me. Listen to me, please. This is the experiment. We want to understand which type of replication is correct. Is it, for example, in this slide, is it dispersive? Is it semi-conservative? Is it conservative? We didn't know, okay? We had this experiment to understand which type is correct. At first, we culture the E. coli in nitrogen 15. It means the older DNA has the nitrogen 15. So the older DNA has nitrogen 15. And then they transfer it to nitrogen 14. It means the new DNA had nitrogen 14. Okay? After one cell division, they extract the DNA and transfer it to the centrifuge gradient, and they understand that we have one bond combination of 14 and 15. It means one bond combination of new and old DNA. And after two cell division, they put it in centrifuge gradient. It means we have one bond combination of new and old and one bond new DNA. In 4 minutes, use the semi-conservative, but how? Yes, but how? Separation of two strands, provide two templates. The card. Okay, listen to me. Listen to me here, please. Listen to me here. If it was conservative, listen to me. If it was conservative, what does it mean? It means from one old DNA, we have one new DNA. So, how many bonds should we saw? Should we see? We should see one. Old DNA, it means one bond for 15 and one bond for 14. Did we saw two bonds in the first extraction? Did we saw two bonds in our first, look at this, we had one bond, yes? So from this understanding, it means that conservative is incorrect, yes? If the conservative was correct, two bond, but we see one bond, yes? Understand? Everybody understand up to now? Okay, thank you. So let's go to dispersive. Dispersive means one part new, one part old, one part new, one part old. It means after the first replication, we should see one bond combination of 14 and 15. Did we saw one bond in the first? Did we saw one bond? Yes. So now it doesn't deny in the first replication, it doesn't deny dispersive. But what does it happen in the second replication? Look at this. In the second replication and in the second cell division, also we have one part old, one part new, one part old, one part new. So how many bonds should we see? Just one. Combination of 14 and 15. Yes? If it was dispersive, how many bonds should we see? One. Combination of 14 and 15. Combination of new and old. Am I right? But how many bonds did we saw? How many bonds did we saw? Did we see? Two. So, is it right? Is it right? No. Yes, thank you. So the dispersive isn't correct too. And semi-conservative have one bond combination of new and old. So we have one bond combination of 14 and 15. One bond combination of new and old. Is it right up to now? Yes. Yes. Okay. After the second replication, after the second cell division, listen to me. After the second cell division, we have two bonds. Look at this. One is combination of old and new, and one is new. So, what type of replication is true? Is the semi-conservative. Understand? Okay, thank you so much. Thank you so much. Okay. Listen to me. For DNA replication, our DNA is two-strand double helix. Yes, am I right? Our DNA is two-strand double helix. But if the replication starts, we have some supercoil in the structure of DNA. Do you know supercoils? Do you know what supercoils mean? Supercoils. Okay, so if replication starts, we have some supercoils in it. So if we have so many supercoils, can the replication continue? No, because the DNA is so condensed and the replication can't continue. So we have some kinds of enzymes. We call them topoisomerase. We call them topoisomerase. Do you know about topoisomerase? Do you know about topoisomerase? Okay, thank you so much. So the topoisomerase can convert supercoiled DNA to relaxed DNA. Relaxed DNA. So they can convert supercoil to relax and also relax to supercoil. So what does supercoil do? Supercoils break one strand of DNA, can break one strand or double strand of DNA and turn it and connect it to each other. So look at this. They open one part of the DNA. You can see they can make supercoil to relax. Okay, thank you. So look at this picture. Look at this picture. When the replication fork go through, when the double strand breaks and the replication starts, we have replication fork. Replication forks go through this way, and after that we have supercoil here. Yes, we have some supercoils here. In this type, we have to use some topoisomerase, it's better to say, topoisomerase break one strand and connect it to each other, so we can see we have open DNA. You can see the pictures? Okay, Mr. Ali, please refresh your site. So, anytime, listen to me, anytime the replication starts and the replication fork go through, in the above side, we have some supercoil. Which enzyme remove these supercoils? Tell me, please. Which enzymes remove supercoils? Tell me, which enzymes remove supercoils? Topoisomerase. Thank you, Mr. Haydar. Thank you. So, in the replication, we have some supercoils above the replication fork, and the topoisomerase remove the supercoils and let the replication fork to continue. Okay, thank you. Okay, I just want to tell you about the loop replication. This is the loop replication. Do you know about it? About this kind of replication? Did you study this?
not rolling circle. It's D loop. Okay. Listen to me. In mitochondria, Mr. Haidar, in mitochondria, the replication type is D loop. Listen to me. At first, in one part, in about 500 base pair, the DNA separate. You can see this blue and primer synthesize. So, at first, in one part, about 500 base pairs, two strands of DNA separate. After that, RNA primer synthesize and replication from one strand start. After it comes to three-fourths of one strand, another strand starts replication. So I repeat it one more. Listen. D-loop replication is a type of replication which occurs in mitochondria. Okay? At first, in the place of D-loop, we have some place which is 500 base pair, two DNA strands separate. We have one primer, which is RNA, one primer synthesize, primer continue replicating, and after its length is three-fourths of the first strand, the other strand start replicating. So tell me please, after one strand finish, the replication, what part of the second strand replicates? After one strand finish, what part of the second strand replicates? 1/4. 1/4. Do you want me to repeat it more or understand? Okay, it's so easy. Listen, at first, one part of DNA, about 500 base pair, separate, and one primer synthesize. After that, one strand continue replicating. After it replicates three-fourths of the first strand, the second strand starts replicating. First time I hear about mitochondrial DNA. Really, Mr. Haidar? let me ask you a question. Anybody, please answer me. Do you think no, let me ask you like this. About the portion of genetics which mother and father give to their children. Okay, do you think mother gives the children more genes or father? Why mother, Fatima? Do you think mother gives more genes to children? No. Mr. Haidar, the sex determination is from father. The sex determination is from father, so it means the mother doesn't have any role in sex determination. It means that if the man, no, don't be sorry, I just want to explain for you to understand. If the man give the Y sperm, Y sperm to his wife, the wife will have boy. Yes? But if the male give the X sperm to his wife, they will have girl. So the sex determination is from father. Understand? Yes. So the father has X and Y. So the father determines the sex of the children. Okay. After that, how many chromosomes do we have? How many chromosomes do we have? We have, Mr. Ali, we have 46 chromosomes. 46 chromosomes, yes, which we have 23 from our mother and we have 23 from our father. Yes? So now they have same role. 23 mother, 23 father. But we have mitochondria, DNA too. How can we get our mitochondria DNA? We can give it from our mother. Yes, understand. So the mother has more role in our genetics of father, mother. Why? Because we take our mitochondria DNA from our mother. Understand? Okay. So I told the male students, be careful about choosing the correct wife. Yes, because your wife give more genes to your child, to your children. Genetic is very interesting lesson. As you know, my field is genetics. It's so sweet. I hope you understand and learn it too. Yes, I told you. We have 23 from mother and 23 chromosomes from father. After that, we have additional mitochondria DNA from our mother too. Understand from now? Do you have my voice? Okay, let's go to study about rolling circle. I was talking about rolling circle before. Do you remember? Do you remember? So, this is rolling circle. Rolling circle replication occurs in phages and viruses. So it occurs in phages and viruses. I told you at first we have one break in one strand of DNA, the new nucleotides attached to the 3', the inner strand rolls, and the inner strands play a role of the template, and the outer strand continue like this. After that, I told you it become double strand. After the double strand, what has happened? It will break in different parts and it become circular. Okay. Is it clear? Cast site. Yes, Bushra, thank you so much. Yes, thank you so much. It was cast site. In Landafage, it was cast site. Thank you so much. So, what about the speed? This kind of replication is slow or fast? This kind of replication is slow or fast? Bashra. Oh, okay, thank you. Thank you. I'm so sorry. Fast. Yes. It is very fast, I told you. This kind of replication is so fast because the viruses should replicate very fast. Okay, any questions? No. Why? Because for example, phages or viruses, they infect one cells. No. I mean the whole process of replication is too fast. This is I told you before. We have so many products in the less time. This is so fast replications. So, for example, if we have bacteria and we have one phage on the surface and we have some DNA of phage in the bacteria, they replicate so fast and after that they will lyse the bacteria and come out. Understand? Okay, let's talk about replication of bacterial chromosome. Yes, Lytic. Let's talk about replication of bacterial chromosome. In the bacterial chromosomes, look at this. the replication starts from origin site. So the replication starts from origin. So we have one part which is origin. What about the replication, the fork direction? We have two fork direction. One is this side and one is this side. So in the bacteria, the replication starts from origin and it will go like this. One this side and one this side. Understand? This is the direction of replications. Do you know how many origins do we have in eukaryotes? In eukaryotes, as I told you, we have one origin in bacteria, in prokaryotes. But can you tell me how many origins do we have in eukaryotes? I told you where does the replication start? Origin. Am I right? How many origins do we have in prokaryote? One. So we have one origin in prokaryote. I asked you, how many origins do we have in eukaryotes? How many origins do we have in eukaryotes? So we have one origin in prokaryote. Is it right? Yes, one in prokaryote. But we have multiple origins in eukaryote. Why? Because the genome of eukaryote is so long, so bigger than the prokaryote. So if we have just one origin, we couldn't replicate all the DNA because their genome is much bigger than the prokaryotes. So in eukaryote, we have multiple origins, but in prokaryote, we just have one origin. Understand? Okay, I told you because the genome of eukaryotes are much bigger than the genome of eukaryote. For example, yeast or human. Okay, the genome of human is much bigger than the prokaryotes. Yes. So if we have just one origin for replication, if we have just one point which start the replication, we couldn't replicate all the genome. Yes? So in eukaryotes, we have multiple origins, multiple points which replication start. But in the prokaryote, we have just one origin. Understand? Thank you. For example, in humans, every 150 kbs, we have one origin. And in yeast, every 36 kb, we have one origins. So in our body, in the human, every 150 kb, we have one origins. But in the prokaryotes, we just have one. Understand? What is the, how about the direction of replication fork? The direction is, you can see, like this. The direction of replication is two-side. Direction of replication is two-side. Two-side. Okay. Look here. One comes like this. The direction. One. The replication starts. The direction. Yeah. The direction. So sorry, it doesn't come up to now. And this is the another direction, comes like this. Okay, so we have one origin, two direction of replication. So this is very important. Listen to me. In direction replication, we need primer. I will tell you, yes. Every time the replication starts, we just want a primer. Okay, I will continue in the further. I want to talk about the structure of origin. Listen carefully, very important. I want to talk about the structure of origin. So, I want to talk about the structure of origin in E. coli. The structure of origin in E. coli. We call it ORC. Okay, ORC. Listen to me. Listen. We have 5 motifs. We call it 9 nucleotide motif, or we call it 9-mer. 9-mer means 9 nucleotide. Okay, so we have 5 domains, which is 9-mer, or means it has 9 nucleotides, and we have 3 domains, which is 13 nucleotides. We call it 13-mer. 13-mer. It means 13 nucleotides. So listen to me. In the structure of origin of the E. coli or ORC, we have 5 9-mer domain, and we have 3 13-mer domains. Okay, so what is the function of 9-mer domains? We have one protein, we call it Dna A. Dna A connect to these 9-mers. Dna A connect to these 9-mers. How many Dna A proteins? About 30 proteins of Dna A connect to these sites. Okay. And what is the structure of 13 nucleotide? 13 nucleotides is 80-rich domain. 80-rich. Can you tell me what does it mean when I tell you 80-rich domain? It means we have so many A and C nucleotides. If the domain is 80-rich, it means it has so many A and C nucleotides. Okay? So tell me please, if we have one part which is 80-rich, we have so many 80 in the domain. How is the strength of two strands of DNA? A and T has 2 hydrogen bonds. C and G has 3 hydrogen bonds. Yes? If we have one domain which is AT rich, how is the strength of two strands of DNA? Is it weak or very strong? Understand me? Look, if we have one double strand of DNA, which is A and T, how many hydrogen bonds for A and T? How many hydrogen bonds? We have 2. Yes? We have 2 hydrogen bonds. Yes? What about if we have 2 strands of DNA, which is CG domain? CG domain. How many hydrogen bonds? 3. Yes. Which is more strong? Which bond is more strong? Which bond is more strong? 3. It means CG. Yes? If we have CG domain in our DNA, our bond, our hydrogen bond is more strong. Yes? Yes. So listen to me. If we have AT rich domain, what does it mean? It means they have weak interaction. Yes? So the strength is less than CG rich. Understand? So I just want to repeat more. In the structure of origin of E. coli, we have 5 9-mer. We have 5 9-mer parts, which DNA attached to them, and we have 3 13-mer, which is AT rich. I told you in the AT rich domains, the strength of double strand DNA is so weak, so they can easily break, they can easily separate from each other. Okay? So after DNA are attached to 9-mer, look at this. I told you 30 DNA are attached to 9-mer. DNA fold around the barrel of DNA A proteins. It will force this region. In this region, our strength was so weak. We have AT rich domain. Yes, it will force this region and the hydrogen bond will break. So 2 strands separate from each other. Understand? So I will tell you one more quick repeat. Listen to me. We have 5 in the origin of E. coli. We have 5 9-mer domains, which DNA are attached to them. Listen to me. We have 5 9-mer domains with DNA are attached to them. After 30 DNA are attached to it, DNA will get around the DNA barrels, and it will force 13 mer domains. What about 13 mer? 13 mer was 80 reach. 13 mer was 80 reach, which the hydrogen bond is so weak. So after the DNA fold around DNA barrel, it will force the 13 mer domains, which the strength of hydrogen bond were so weak, so the DNA separate. Understand? Okay, thank you. After that, we want to helicase to come. We want to helicase to come. After that, our helicase, what does helicase do? Do you know? What does helicase do? Can you tell me, please? What is the function of helicase? What is the function of helicase? Separate, yes, thank you. Separate two strands of DNA. So after that, the helicase, DNA helicase come. Thank you, Roya. Thank you, Haydar. Thank you, Fatima. The DNA helicase come and separate two strands of DNA with the help of DNA C. With the help of DNA C. So, listen again. First, we have DNA A attached to 9 mer, 30 DNA A attached to 9 mer domains, DNA fold around DNA A, force the domain 13 mer, the 13 mer strength was so weak, they separate. After separate the DNA B comes, DNA B is helicase, it needs energy with the help of DNA C. Six protein of DNA C help DNA B helicase to separate two strands of DNA. The direction, listen to me, the direction of DNA B is from 5' to 3'. The direction of DNA B is from 5' to 3'. You can see it in the picture. The direction is from 5' to 3'. So, in which strand, leading or lagging? It continue. Leading or lagging? What is this strand? Leading or lagging? You know this? You know this? Leading. No, it is lagging strand. Lagging strand. It is lagging strand. So the DNA B moves on lagging strand. The the direction is 5' to 3'. So I told you when the DNA it means when the helicase open to a strand of DNA, it forms replication fork. In the above side, what will happen? In the above, we have supercoils. Yes? So in the above, we have supercoils. Which enzyme removes supercoils? Which enzyme removes supercoils? Yes, topoisomerase. Topoisomerase remove supercoils. So like this, after that, helicase B continue separating two strands of DNA, and topoisomerase in this remove supercoils and let the DNA B to continue its function. Understand up to now? Yes, okay. So after, listen to me, after two strands of DNA opens, if you open two strands of DNA, they will like to attach to each other. They will like to become double strand too. So if we open two strands of DNA, if we let it like this, they will like to attach to each other and form double strand of DNA. So what does the cell do? The cells have one kind of protein which name is SSBP proteins. What does SSBP do? SSBP attach to single strand DNA and doesn't let the single strand to attach to each other and form double strand. SSBP is single strand binding protein. Understand? So after the helicase open two strands of DNA, if we let it like this, they want to again form double strand of DNA. But SSBPs come. What does SSBPs do? SSBPs attach to single strand DNA, and after they attach to single strand DNA, they doesn't let the DNA to attach to each other. Okay? Understand? Okay. Thank you. Let's talk about leading and lagging strand. Listen to me. Listen. After two strands of DNA spreads, the replication should start. In one strand, we have primer, which is made from RNA, we have primer. After that, the DNA is synthesized. I told you what is the direction of DNA replication in the beginning of session. I told you the direction of replication is from 5 prime to 3 prime. Yes, Mr. Haydar, 5 to 3. So, DNA is anti-parallel. Anti-parallel. It means if here is 3 prime, we have 5 prime here. And if we have 5 prime here, we have 3 prime here. Yes? So, listen to me. If we have 3 prime, we have 5 prime here. I told you the direction is from 5 prime to 3 prime. Yes, like this. So, at first we have primers and then replication continues. And after that, the replication forks open too, and the replication continues like that. For example, we have like this. This strand continuously continue the replication, so we call it leading strand. What is leading strand mean? The leading strand means the strand which continuously replicate DNA from 5 prime to 3 prime. So the strand which is continuously synthesized, we call it leading strand. And because this is 5 prime, we have 3 prime here. The direction was 5 prime to 3 prime, yes? 5 prime to 3 prime, like this. We have 1 primer here, 5 prime to 3 prime. Again, 5 prime to 3 prime. So, one strand synthesized partially. One strand synthesized partially, and we have some fragments. We call them, what do we call them? Do you know? We call each fragment Okazaki fragments. We call them Okazaki fragments. Did you know? So, we call each fragment Okazaki fragments, and after that, these Okazaki fragments connect to each other. We call this lagging strand. We call this lagging strand. So, just listen to me. Just listen to me. So, one strand continuously replicate. We call it leading strand. We have one primer in the beginning. And one strand, because it should synthesize a 5 prime to 3 prime, and the direction of the synthesis is opposite of the direction of replication fork, it will synthesize uncontinuously. We have fragments of DNA. We call the fragments Okazaki fragments. Understand? Okay, thank you. So, the the direction of leading strand replication and the replication fork is the same. Listen again. The direction of leading strand and the replication fork is the same. So as the replication forks go, it goes to, so the replication fork direction and the leading strand are the same, so it goes continuously. But the replication forks direction and the replication of lagging strand are the opposite side, so it synthesizes uncontinuously. Okay. I hope you all understand. Yes? Let's talk about DNA polymerase. I told you in the previous session and you asked me. We have 3 DNA polymerase in prokaryotes. So, in prokaryotes, we have 3 DNA polymerase. DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. Okay? What is the main replicating enzymes in prokaryotes? Do you know? What is the main replicating enzymes in prokaryotes? The main replicating enzymes is DNA polymerase 3. So DNA polymerase 3 is the main replicating enzymes in prokaryotes. Okay, so I told you what is the function, what's the direction of replication 5' to 3'. So look at this. We have some exonuclease activity. In the DNA polymerase 3, we have 3' to 5' exonuclease activity. Look, we have 3' to 5' exonuclease activity. What does it mean? It means if the direction of replication is 5' to 3', it has the 3' to 5' exonuclease activity. It means it can remove from 3'. So what is its point? If the DNA polymerase put the wrong nucleotide here, if the DNA polymerase put the wrong nucleotide here, it can remove it from the 3' end. It can remove it from 3' end and correct it with the right nucleotide. So we call this function proofreading. Understand? Do you want me to repeat? Okay, listen more, Fatima. Listen, please. It has 3' to 5' exonuclease activity. What does exonuclease means? Exonuclease means remove nucleotide. Okay, from where? From 3' to 5'. From 3' to 5'. Look. For example, this is 3', yes? For example, DNA polymerase put one wrong nucleotide here. It can remove the nucleotide from the 3' and put the right nucleotide and continue. So if the direction is 5' to 3', and if the DNA polymerase put the wrong nucleotide, it can remove it from 3' and put the right nucleotide. Okay? So we will call it proofreading. What do you call proofreading in Arabic? What do you call proofreading in Arabic? Orhan. Okay. Yes, the correction. So DNA polymerase 3 can correct itself. How? By 3' to 5' exonuclease activity. Remove from 3', put the right nucleotide. Okay, Fatima? Thank you so much. Thank you so much. Okay. So, listen. Listen. What about DNA polymerase 1? Let's see DNA polymerase 1. DNA polymerase 1 has DNA polymerase 1, listen, has 3' to 5' exonuclease activity. I told you 3' to 5' exonuclease activity led the enzyme to proofreading. Led the enzyme to proofreading. It means let the enzyme to remove the incorrect nucleotide and put the correct nucleotide again. Okay? So if the enzyme, if the polymerase have the 3' to 5' exonuclease activity, it means it can correct itself. It has the proofreading activity. So tell me, DNA polymerase has the proofreading or not? Tell me, does the DNA polymerase 1 has the proofreading activity? Yes, yes. Prime and correct it. So if enzyme have 3' to 5' exonuclease activity, it has proofreading. Okay. Thank you. If enzymes have 5' to 3' exonuclease activity, look at here, DNA polymerase 1 has 5' to 3' activity. What does it mean? Look at here, 5' to 3' exonuclease. It can remove from 5'. It can remove from this part. So what does it mean? It means it can remove primer. Yes? Look at here. DNA polymerase 1 can remove from 5'. So if it can remove from 5', it can remove primer. What does primers made of? Primers, as you know, are RNA. Yes,
We shouldn't have RNA in our DNA, so they should be removed. How? RNA polymerase 1 remove the primers with the 5' to 3' exonuclease activity. Understand? So, I will repeat more. Listen. By the activity of 5' to 3', DNA polymerase 1 can remove the primer from 5'. The primer made of RNA, they should be removed. So, RNA polymerase 1 remove the primer and fill the gap. So, we will say DNA polymerase have the gap filling activity. Gap filling activity. What does it mean? It means it can remove this primer and fill it with DNA. Fill it with DNA. It can fill the gap. Understand? And what does the function of DNA polymerase 2? DNA polymerase 2 mainly activate in repair. DNA repair. So, DNA polymerase 3 is the main replicative enzyme. It has proofreading, but it can't remove the primer. DNA polymerase 1 has proofreading activity and can remove the primer, fill its gap. We call